Okay. Regarding the good morning first, everybody. And regarding the final exam, I want to make it clear for one more time that final exam is going to be more difficult than the midterm. In terms of the magnitude, the midterm is much, I would say, 2 times easier than the final exam. Okay. So which brings me to the nature of the questions that your colleague just asked. What is the nature of the questions that will be used? Substantial amount of the exam is going to be multiple choice. However, the multiple choice will be more challenging compared to the ones that you've seen in the midterm. In addition, there will be some short question answer, so you have to write down or discuss certain aspects, and I will give you some examples as I go through the class today. Then there will be some like I can't say exactly how it looked like the question unless I give you an example of it, like signaling pathways, description of apoptotic, for example, pathway, downstream effects of reactive oxygen species, stuff like that, where you have tons of material or images or case studies that we discussed that you would need to write down yourself. So, which means you have to get prepared in a different way compared to what you have prepared before for the midterm. Because when you have the multiple choice question in front of you, you have the answer. One of the 4 or 5 choices is an answer, which if you were a little bit confused, then you try to remember it, you will find the answer in the multiple choice. However, when I say, for example, describe the apoptotic pathway downstream fast ligand, which means an extrinsic apoptotic pathway. This means you have to remember that. You have to say that signal bind to the receptor, that receptor get activated, that receptor get phosphorylated, this protein get phosphorylated, and then depends on what the question I will be asking. But you have to be prepared that now you are integrating more than one thing. And I kept saying this right after the midterm. You have to get prepared much better for the final exam compared. So please don't take it easy like you did in the midterm and you think the exam is going to be that easy. Just make it sure for you guys. Now, another thing I really noticed, I'm not accusing anyone, but I really noticed that many of you don't read their emails, don't read the announcements. And I will give examples. Assignment was an example where we said clearly, if you want to do your assignment on your own, that's fine, but you still need to register and go to a group so that we know which disease you are discussing. Many of you didn't do that. And I sent announcement 1 time, 2 time, emails 1 time, 2 time. So if you guys didn't do that, I'm sorry, you didn't read your email, you didn't register to the assignment. Then I can't help you if you come at the time of the submission and you don't know I don't know which group you were working at. So that's why some of you were penalized for this. So this is an example. Another example is last week, I had a meeting and I moved the office hours from Thursday to Friday. And unfortunately, there's only one person that showed up. The week before, it was regular office hours. There was only one person who also came up. And I'm opening the discussion for you guys every time that if you have questions, please let me know. If you have gone through the material and it's more difficult than you think, come back to me and let me know so I can go again on this material. But surprisingly, no one asked anything. So, which tells me either what I'm giving you is super easy for you, which is great, and I'm really happy, or you are not really digesting the material yet or delaying it, which is a problem. So, uh, I'm just giving you a hint, and exam is in 4 weeks, so there's not too much time left. Okay, so I have a note. I am trying with my okay. The exam has to cover everything because we can't really split the concepts. So your colleague is asking here, um, your colleague is asking if the exam is going to covering all the material. Yes, the final exam is going to cover all the material because all the concepts we described, we are reusing them to understand the disease. So we can't really delete any of those material. Margaret, yes, I'll try to do that. I don't promise that I can send you those. I do have many other things, and I'm really locked in time when I can make these slides ready. Yes, next week you will have a quiz. Uh, you will have sample questions. I can't really post a quiz for each section by its own because it will be like probably extensive. I wouldn't have material to do for the final exam. But next week you will have the quiz, and then you will see the quality of the questions, how it will change. The bottom line, if you are really reviewing the material, you don't have to really be worried. But if you're not reviewing the material and keeping them later on, there's lots of details in these slides. Myself, when I read those slides again after the class, many times I go and review how much time I spend on each slide. I found some of them difficult. So I don't know how you guys finding it that easy and nobody is asking questions. I will give you an example, okay? So let me share my PowerPoint with you, okay? So let me go quickly to one of these slides that we had probably 3 times now. So let's do this. For example, who can tell me what second mitochondrial-derived activator of caspases, or what is called SMAC, does? And who can tell me what is inhibitor of apoptosis, or IAPs, does? So quickly. Anybody is ready to answer this question? I don't want you to tell me what it binds to, what it activates, just the general name, the general, what exactly those two proteins do. What's the function of SMAC and AI, IAP proteins? Just general, what's the function of these two proteins? Very good. Very good. Very good. There you go. Some people already did the homework. That's great. Okay, so I will give me just 1 second to hook my computer to the socket so that doesn't need battery. Sorry about that. It seems that the connection has been lost and I just recovered it. So I'll restart. Could you please tell me you see my screen? Okay, cool. Thank you. Thank you. Now with this starting, okay, I shall cancel. Okay, so we have the recording. Let me make sure. Okay, cool. So today's topic is gonna finish up on what we have discussed in the previous lecture, where we started discussing the signaling pathways and how the signaling pathways get to changes into the cellular function. So as you can see in this slide, this is, I would say, it's a simplified circuit of how the cancer prevails. And although it looks very, very complicated because there's too many intermediates, if you look carefully, you will see that we have already covered a lot of information about that. For example, we discussed how P21 under the control of P53 could lead to a process of cell cycle change. And that could be also a signal that goes side by side with the cyclin Ds. So which in principle affects other genes like the phosphorylation of retinoblastoma protein and then changes in the gene expression in general. So that was in lecture 3-4, I think, when we discussed that. Another aspect that we really have discussed in terms of the apoptotic survival and death pathways. So survival pathways are mainly maintained by BCL2 family, which sequester the apoptotic partners of these proteins. So there's a balance between the BCL2 protective or compared to those who are pro-apoptotic. Today, also we discussed in the previous class the tyrosine receptors and how the tyrosine kinase receptors lead to a propagation of a signal that comes from a growth factor to proteins like GTPases and how the signal propagate into a secondary messenger. With these circuits, it's just going to show you, well, obviously I'm not going to use this, for example, for the exam because it's very complicated. But it's showing you that while in some of our classes we were focusing on only one aspect, like here P53, the tumor suppressor, P21, and cyclin E, this is happening simultaneously with all the other signaling pathways. And the feed-in of, for example, death signal and a survival signal could happen at the same time, as well as cytokines. So this is giving you a snapshot of what the cell is performing when it gets multiple types of signals. Okay, and why these are important to look at, it's because when we speak about cancer, we find out that most of these signaling pathways are altered, and altered in a way that are probably just opposite to how they used to work in normal cells. So, for example, if we take this one here, because we discussed it in a previous class, so if we have anti-growth factors, or for example, loss of nutrients. So this cell is probably, or mutations, like there is a DNA damage. In all of these three situations, the cell has to stop growing, right? It has to stop dividing. So one thing for the cancer cell to continue growing is to block this signal here. Therefore, it doesn't respond to anti-growth factors. And similarly to this one here, if it receives a death signal, it would have the ability to sequester the pro-apoptotic signaling, and therefore it can escape apoptosis. Similarly, survival factors are needed, and therefore cancer cells probably can produce them. So they can be produced by the cancer cells, and then they could affect the cell. So that autocrine effect that we discussed last time. And obviously, when we discuss the signaling, there are typically 3 or 4 types of signaling that we always find that being altered in cancer. Obviously, I don't want you to know all of them. I just focused on a couple of them, which is the RAS and the PI3 kinase. Okay, so this is how the lecture is going to go. Again, office hours, please come whenever you need. So we will discuss the characteristic of the cancerous cells, the causes of the cancer, the types, the tissue morphology, how it looks like, and we will touch on the diagnostics and cancer treatments. Okay, so before we proceed, last time we have discussed the uh, G proteins and how the signaling take place within the G proteins. And then we discussed the tyrosine receptors, and we took insulin as an example because it binds to tyrosine receptors, and tyrosine receptor kinase is being activated downstream. Now, in this lecture, we still need to know the third signal transduction pathway, which is functioning through the serine threonine kinases. And there's not a lot of difference in terms of how the signaling take place. And they are more or less the same. But here you are talking about a phosphorylation event that takes place on the serine or the threonine instead of being taken on the tyrosine. Obviously, this is a selective pathway, so the downstream signals could be different, and therefore the second messengers in general could be different. Now, when you study cell signaling, keep this in mind. When you study cell signaling, provided that the cells, they have similar receptors and signal transduction pathway, the signaling pathway could be different outcome from one cell type to another. And this can be easily understood because the downstream targets or the downstream effectors are different from one cell to another. So, for example, I given here three different examples from T cells, which are the thymus educated version of the immune cells. And it's the T cell receptor that we are looking at, which triggers the phosphorylation of the serine threonine kinases. So if you look from the previous class, we discussed that this molecule, the phosphatidylinositol diphosphate, which is recruited to the membrane with these protruding hydrophobic tails, is most of the time phosphorylated to become phosphoinositol triphosphate. Okay, now the activation of this form of the molecule leads to its release from the membrane, and then it would lead to the activation of the downstream kinases. Yes, Margaret, I'm going to repeat that. So, and this would lead to the activation of the downstream targets, which in this condition could be PKC, which is a protein kinase C. And this propagates more and more as you go. One kinase has multiple downstream targets. And for example, if we took this axis here, the PKC and PKD2, you will see that we have secretion of interferon gamma, interleukin 2. And those, they are needed for an immune response. Remember, the release of these two proteins could activate neutrophils, could activate different immune cells as well. So just to get back, so this slide is not very well detailed, Margot, because in the previous slide, it was much more detailed how the phosphatidylinositol diphosphate becomes triphosphate. So let me repeat, so probably to refresh the memory of those who didn't remember. When the signal gets to the receptor, the name of the receptor is different from one cell type to another. And if I will present, probably I will send you a slide for further reading of the how many types of receptors we have, and to remind you how the activation takes place. But generally speaking, when a signal binds to a receptor, there will be some conformational changes and causes the internal part of that receptor to become phosphorylated. And when this part becomes phosphorylated, then it has an ability to phosphorylate other proteins within the cell. And you can tell that in terms of organization here, there's the receptor and there's the phosphatidylinositol diphosphate, which is just close to it. So therefore, whenever this receptor becomes active, it will directly phosphorate this protein here. And don't get confused too much by the name phosphatidylinositol diphosphate. It's just at the residues 4 and 5, and now you added one more phosphate, so it's become phosphatidylinositol 1, 3, and 4, so triphosphate. Okay, then the downstream targets are becoming activated. So I want you to digest this part very well. Okay. Now, as we go more in the discussion today, you will see that there are more slides that will describe for you how the activation of the PKC take place or other downstream targets. Okay. So many times, many times the names that are written here, that are written in the graphs or in the schemes, they are described here. So you have more text to read what is those. And yes, so for example, I could ask you a question where I can present this image for you, and I could take out PKC and PKD out. And I would ask what are the downstream signals between this guy and that guy. So that's one of the questions. Now, I didn't mention anything about these guys because they are very specific for the T cell activation, and I don't want you to remember those because you will forget them for sure. But the signaling pathway, it goes through these set of proteins, and this is something you need to know. Now, many times I mentioned this, if I don't give you the text, you will be asking what is this MTOC, and if I give you the text, you ask me if I ask you about it in the questions in the exam. So fair enough. Sounds good. That's why I'm here, so to make things clear. So if my computer wants to go to the next slide, which is not the case. Just please be with me a minute. My computer is frozen. Okay. So this is one target. So this is one arm, sorry, of the serine threonine signaling pathway, which in principle leads to the generation of diacylglycerol, right? We discussed that when this is being phosphorylated, and please go back to the previous lecture. I discussed clearly how that happens. You will have the generation of the diacylglycerol, which plays a role in the activation of the phospholipase gamma here. So this was from the previous class. And remember that the diacylglycerol also plays a role in the signaling of calcium, so because it can translocate to the ER. If you have confusion about this, please go back to the previous class. Now, the second arm of the serine threonine kinases is the RAS. So this is RAS-regulated serine threonine kinases. So this means whenever you say the protein RAS, it is most of the time recruited with GTPase and GEFs. So in this slide here, I'm focusing only on the GTPases because for RAS to become active, it needs GTP, okay? And there is an important aspect that we should not forget that if its signal comes through the receptor and activated like you see the acylglycerol comes from the phosphatidylinositol 2 to 3, right? And now we have that. And this is a signaling cascade that induces RAS activation. Now, RAS activation requires energy, means requires GTP, which is the molecule that we were producing from our nutrients, right? When the GTP is bound to RAS, then RAS is active. And now RAS could induce downstream signal like RAF, MEK, and ERK1 and 2, which are going to be coming our most important or the second important in cancer related signaling. So, but don't forget that the cell, once it gets this signal, it has at certain point to shut it down, because the moment that the signal gets in, it gets this receptor phosphorylated and the signal is internalized. Those are the second messenger. You don't want this to be turned on forever. You want at certain point to turn off that signaling, because if you turn it on forever, it's going to be destructive for the cell. And that's why these proteins are important. The RAS GTPases are very important, because the RAS GTPases, those are the ones that regulate how long the RAS becomes active. And they can switch the RAS like from inactive state to an active state. And then their counterpart, which is called GIFs, which are presented, I think, here, RAP GEFs here, GEF. Those are the one that brings RAS back to the normal. And therefore, yes, you get the signal, you activate this pathway, but this is not forever. That's the main point I wanted you to understand here, that while the signaling is taking place in RAS, it depends on the GTPases, RAS GTPases to hydrolyze and activate the RAS, while the GAFs needs to get this back to the active state. Now, downstream signals, it's nothing really new for you that this protein is going to phosphorylate that, and this protein is going to phosphorylate that, and now this is active. So it's all like a cascade of phosphorylative events where the phosphatic group is being added to these proteins. And remember, adding a phosphate is adding energy. And probably adding that phosphate is also changing the conformation of the protein a bit. So it makes it more accessible or ready for activating its target. And therefore, if you keep going through this cascade, the downstream elements, which you see, for example, here, the initiation elongation factor 4E, it plays a role in the translation process. So you could tell that this signal is gonna induce translation already. And in all these schemes, probably it skipped my mind to be like tell you carefully that whenever you have this arrow, it means it's an indication of activation. And whenever you have a blockade, which is not here, probably it will come in the next slide, it would mean that the protein is deactivated. So in this second type of serine threonine kinase signaling pathway, you will have the diacylglycerol as the initial second messenger, which activate the GTPases. So therefore, now you have the RAS being bound to the GTP, and downstream that you have the activation of RAF, MEK, and ERK, and this is the MAP kinase. So the RAF, MEK, and ERK1 and 2, this is the MAP kinase, okay? Now, ERK1 and 2, these are kinases that play a role in multiple cellular processes. So I listed some of them here. Cytoskeleton, cell metabolism, transcription, translation, and proliferation. So this is like 5 major different components that these ERK1 and ERK2 complicate. And if I will show you more, it will become much more complicated. So I'm just touching on this. Yeah, these are the downstream targets of the of the MAP kinases. Yes, Margaret. So the third is the phosphatidylinositol triphosphate itself. And phosphatidylinositol triphosphate itself could act like a docking station to other kinases to come and bind and get phosphorylated. Now, this slide, I really want you to know very, very well this part here. I don't want you to know the difference between these two parts, the mechanism 1 and mechanism 2, but I really want you to know the PI3 kinase AKT pathway very, very well. If you look for AKT, or some people call it ACT here, or protein kinase B, all these names are like similar, like for the same protein. Now, this protein plays a central role in many, many, many functions. And today we will be looking downstream, as we discussed in cancer, how PI3 kinase and AKT play a role in making the cell cancer, the cells more proliferative, more surviving, and less prone to cell death. Okay, so bear with me. This slide is a little bit busy, but let's go back to where we started in the previous 2 slides. You got the receptor, the T cell receptor alpha beta or CD3, whatever its name. It's a serine threonine kinase way of signal transduction that now the activation of the receptor to get the phosphatidylinositol diphosphate to become phosphatidylinositol triphosphate. One second. So the transfer of this diphosphate into the triphosphate needs a kinase activity. Okay. So the kinase that performs this is called PI3 kinase, which is again phosphatidylinositol 3 kinase. It will simply add a phosphatic group and causes the phosphorylation to make the triphosphate. Now, how does this become active? It could bind to the receptor itself, because now if the signal comes here, and I have a couple of videos for you to watch about that, and there's a little bit text as well. So when the receptor comes, where the signal comes and binds to the receptor, and the conformational changes happen, PI3 kinase, which is flowing in the cytoplasm, can come and bind to the receptor. That's how it becomes active. And then it can go into the phosphatidylinositol diphosphate and leads to its phosphorylation, which now we have phosphatidyl triphosphate is active. At this point, AKT can get recruited to that one and get phosphorylated. Okay, now AKT has 2 sites of phosphorylation, as you can see here. One of them is the threonine 308, and the other one is serine 473. And as you can see from the title of the slide, it's serine threonine kinases. So now we are talking about a protein as a second messenger that has recruited that signal from the receptor from the second messenger, which is PI3 phosphatidylinositol triphosphate, into the AKT. Now, which one phosphorylates AKT? There are many theories, many papers, which one comes first, which one comes second, and what types of complexes are made. I don't want you to know that. I want you to know the signaling pathway all the way to the activation of the AKT and then the activation of the sites, the threonine and the SERM. Good. Now, whenever AKT is activated or phosphorylated, now it can phosphorylate downstream targets. So one of these downstream targets are FOXO proteins. And FOXO proteins, these are family of transcription factors that normally reside in the nucleus. Okay. When those get phosphorylated, they will get translocated back to the cytoplasm. And their translocation gets them sequestered with this protein, the 1433, and eventually get them to the proteasomal degradation. Okay, and that's how you could lead to an effect on blocking the activity of the FOXO proteins. Okay. Questions here? Good. So let's go quickly to watch one. So I think we watched already the GTPases and the receptor tyrosine kinase in the previous one. I would watch the PI3 kinase, which is a very simple 4 minutes, I think. Great. And let me know if you see my screen, otherwise I will need to change the, okay.
One of you can create your own website by saying, this is what I want. With Wix, you can.
I don't see the screen. It's probably again. Okay. Okay, so let's see how the PI3 kinase is activated.
Hey friends, and welcome to today's video about the PI3K ACT-mTOR pathway. This pathway leads to the activation of mTOR, which will have positive effects on cell cycle progression, cell survival, and cell proliferation. This pathway is actually often disturbed in cancer cells. Please keep in mind that mTOR has a lot of effects in the whole cell, and in this video I will just touch the basics. So the pathway basically starts with the molecule called PI3 kinase, short PI3K. We can see here that PI3K leads to the activation of ACT, and also here in the side branch we see that there is a protein called PTEN which can inhibit this activation of ACT. PTEN is a very well known and studied tumor suppressor gene, but for simplification we will just exclude it here. PI3 kinase will activate ACT, and ACT can then lead to the activation of mTOR. To name just two main effects of mTOR, we have upregulated cell growth, and mTOR also promotes cell cycle progression due to protein synthesis. How is this whole pathway activated? Here I show you two possibilities. It has been shown that the PI3K pathway can get activated by the RAS GTPase. However, in this video we will focus on the activation of the receptor tyrosine kinases. These membrane-bound receptors can, due to transphosphorylation, lead to the activation of PI3 kinase. For the initiation of this pathway, it is required that we have growth factors. So they will bind here to our ligand binding sites. This leads to the dimerization, so the receptors form a dimer, and one receptor will phosphorylate the tyrosine residue on the other one. This transphosphorylation now allows for the docking of proteins. And one of these proteins, which get activated by the tyrosine kinase, is PI3K. The purpose of PI3 kinase is to transform the PIP2, which is a membrane protein binding two phosphates, into PIP3. The transformation of PIP2 to PIP3 activates PIP3. And PIP3 activates ACT now. ACT can now initiate downstream effects and will activate mTOR. And mTOR, to make it short, will upregulate the translation, so the synthesis of proteins from mRNA. It will further, for example, also initiate the biosynthesis of lipids. So let's have a look at the main effects of mTOR. This is firstly the cell survival. The PI3K ACT mTOR pathway will inhibit apoptosis, so mTOR activation promotes cell survival. Further, due to lipid and protein synthesis, the cell can grow. And the main point is that mTOR activation leads to cell cycle progression. These proteins, which are synthesized due to mTOR activation, are involved in the cell cycle, so the cell cycle can go on. You can already imagine that if the mTOR pathway is disturbed, or let's say upregulated, this can easily lead to uncontrolled cell divisions, which can cause cancer. Lipid, protein, and amino acid synthesis, which is definitely upregulated by the mTOR pathway, can also be summarized as anabolism. In general, the mTOR activation has anabolic effects on the cell. That's it for today. I hope it was very helpful for you. Please keep in mind that in this pathway, there's a lot of crosstalk. My illustration was very much simplified. That means that pathways in the cell can cross-interact with other pathways, and there's a lot of signaling going on. But this is a...
Okay. Okay. So can you see my screen again, please? Anybody?
Yes.
Thank you. Okay, so this provides you with a snapshot how the PI3 kinase gets activated, and now we have two arms that leads to the activation of AKT and then the mTOR. Now, don't get confused between receptor tyrosine kinases and serine threonine kinases, because the serine threonine kinases are in principle those downstream second messengers. Okay, at least in the ones that we have discussed so far. Then there was a question probably before that I forgot to answer, which proteins activate the RAS and which one that deactivated it, right? The proteins that activate the RAS are the GTPases, and those low hydrolyze the, not hydrolyze, those add the GTP and gets out the GDP from the pocket of the RAS. And when the deactivation cycle starts, the proteins that does the opposite, which is the usage of the GTP and the recruitment of the GDP, these are called RAS gaps. Okay, I didn't want to go to the discussion of the RAS gaps because if I tell you how many of those, you will get like crazy about it. It's been like, I would say there is like 27 or 28 of these proteins. Okay. Now, in terms of the RAS activation, we will come back to describe more in the next couple of slides in terms of cancer. So I thought this would be a very good introduction for you in terms of the signaling pathway so far, and then we will come back to discuss how those signaling pathways are affected in cancer, and then you will have a better understanding why we are discussing those. Okay, so which brings us to the subject of today, which is cancer, which have of course it's probably the second leading cause of death, and obviously it's a very challenging type of disease. Based on what we have discussed so far, you can see how many signaling pathways are being affected in cancer, and therefore, if you think about a therapeutic study or a therapeutic strategy, then probably you could affect one of these molecular pathways, but then the others are not affected. And therefore, that explains why cancer therapeutics are not very successful so far, at least not very successful so far. Okay, so the word cancer comes from the racine of a Greek origin, carcinoma, and that explains the shape of that cell with many appendages. It described the growth of a tumor as being a new growth or neoplasm. In principle, you have a normal tissue like you see here, and the description of the cancer came from the fact that there's a growth in this area that is leading to an increase in the size of that organ or tissue. Okay, so it's a cellular tissue description where you have a swelling because of the outgrowth of the cells. But if we look carefully between the normal cell and what's happening in the tumor tissues, then, and in this condition, we are looking at epithelial cells, which are the major cause of all these sarcomas, then the epithelial tissues are very well organized, where those epithelial cells would sit on the basement membrane. And this is probably the lining of all the viscera that we have, like in intestines, in kidneys, in lungs, in most of our tissues in endothelial system. The stroma, which is the basal tissue that is under that basement membrane, then it has many other cell types. Some of them are not really specialized, like the fibroblast, and some others that are really specialized, like mesenchymal cells that could become any other cell type. Sorry, unspecialized mesenchymal cells that could become any cell type. And the basement membrane is supporting those epithelial cells, but then for the stroma, there is what we call the extracellular matrix, the ECM, the extracellular matrix. Okay, it's written here, extracellular matrix. Now, those cells interact one with another, and they keep this extracellular matrix in a very well organized shape. Now, if you look at the image beside when tumor growth starts, first you can see that some of these cells are still normal, relatively normal, and the rest, they start to become deformed. So you can see that there's a differential growth and size. In addition, in the stroma, there is a change in the extracellular matrix. And this is change in the extracellular matrix is part of the microenvironment of the tumor that we will discuss in a few more slides, but I'm introducing it here to introduce you first for the cellular aspect and the changes. So the changes in the extracellular matrix, now you have a more rigid extracellular matrix, so a rigid ECM, which is associated by changes that happened in the cells in the microenvironment. So these cardiac fibroblasts become cardiac fibroblasts associated with the cancer environment. So probably they could react differently to the basal cells. And eventually this interaction is abnormal. Here you can see that these cardiac associated fibroblasts, not cardiac, cancer associated fibroblasts, could send growth signals to the tumor So this is one of the examples. So it's written here for you, fibroblasts and mesenchymal stem cells. They could change the shape, the expression profile, and become more proliferative themselves. And they can secrete high levels of growth factors, cytokines, and chemokines. So at this point, the outgrowth that you have seen in the basal cells leads to that disruption of the basal membrane, and then the changes in the stroma that is associated with the changes of the cell component and the cell function. And that's probably stage number 1, and then that's a stage number 2, where you start having this interaction. Like, I would not say stage, I would say that the step in terms of the ignition of the start of the tumor cells. Now, cancers could be referred to malignant or benign, depends on the their ability to grow and metastasize. And while benign tumors is mostly known that they could only lead to a hypertrophic response, meaning that it's just an increase in the size of the organ without affecting the function, some of them would do. Okay, I'll give you some examples. So remember, we discussed that this is how the cell reacts to growth factors. So if we go back and we discussed that now an epithelial cell that becomes under an viral infect or radiation or a mutation, it leads to its outgrowth and it becomes a tumor. And that leads to the changes in the stroma as well. And then the stroma would respond by releasing growth factors, cytokines, and chemokines. What happens? Those cells will respond to those signals. So some of these signals will be survival signals. Some of them will be preparing the cell to divide, and some of those will ask the cells to differentiate and others to die. Now, it's easy for you to notice that the signals that push the cells to survive are favorable for the cancer, as well as those that are asking the cell to divide more, grow and divide. Not necessarily that the cancer would like to differentiate, because when the cell differentiate is performing a function, so most probably the cancer cell is not going to differentiate. And if they do that, they will do much less level of differentiation. And eventually the cells, the cancer cells, they would not respond to the death signals. Okay, so that explains to you how we go back one step in terms of the signaling pathways. Now, no, these signaling pathways we described, most of them, like PI3 kinase and ERK, those respond to signal, but then they push the cells to survive more and then to grow more. So what happens to those signals downstream? Now, before we go forward, there's just few information for you. Probably you have already known these, most of these. There's nothing really important in this slide. That the major causes of cancer could be divided into 4 or 5 different categories. One of them is genetic, where mutation in certain genes like breast cancer 1 and breast cancer 2, the more the chances that you have a breast to have a breast cancer is that the breast cancer 1 gene and 2 are mutated. In addition to environmental factors, we already discussed UV light, radiations, probably diet, tobacco, alcohol, all that. And you have to link the environmental factors to the internal, like diet and nutrient and reactive oxygen species. Infectious agents, there are a few viruses that are known to increase the risk of cancer. And those are, I will discuss for you here, the human papilloma virus in one of the slides that's coming. Radiation, hormonal imbalance. This is a very good an important aspect that you have to remember, that hormonal imbalance could lead to a chronic activation of a pathway. Remember in the previous class, we described insulin, and insulin is induced in order to bring the blood sugar down, but insulin also induces growth. Okay, and if there is an imbalance in the amount of the insulin, particularly, it will induce the outgrowth of the cells. So if you mix any of those components, like chronic inflammation with hormonal changes or radiation with agents or environmental factor with chronic inflammation, the combinatory effect increases the risk of having cancer. Okay, so the question that I will stop at to take a break after this one is insulin. So I'm just giving you an example of combinatory effects of two factors that could lead to a higher risk of having a tumor development. One of them, for example, increased weight and increased body fat, okay? Now, when we describe the components of the nutrients, how we catabolize those and produce energy, we mentioned that we would produce a lot of fatty acids, and those fatty acids will be source of energy. But if you have tons of these fatty acids because of obesity, then the ability of having some sort of oxidative stress in addition to the system being overloaded with these free fatty acids causes what we call insulin resistance. And the fact that we have tons of those floating around, in addition to other factors that could be inflammatory, that comes with the increased weight and increased body fat, then it would lead to what we call insulin resistance. And the insulin resistance, it's because we have an induction of insulin for a long period of time, because the cells are producing that insulin and being sent to the target cells, and the target cells would react to that by either dying or increasing proliferation. So how does this happen? This happens through downstream signals that are downstream of insulin. So you have insulin receptor and insulin growth factor receptor here, induced receptor. So how does this happen? So let's read it together. So insulin resistance is increased with obesity and compensatory chronic hyperinsulinemia. So you have already a case where you have high insulin that is accompanied by increased obesity. Now, increased level of insulin leads to decreased liver synthesis and blood levels of what? Of this protein, insulin-like growth factor binding protein 1 and 2. So the liver stops producing insulin growth factor binding protein 1 and 2. And therefore, this makes decreased level of growth in insulin growth factor binding protein 1 and 2 induces insulin growth factor 1. When these two factors are down, because it's insulin growth factor binding protein 1 and 2, so these binding proteins are not available anymore, therefore insulin growth factor is more free and more bioavailable. And now insulin growth factor 1 can bind to its receptor and induces the cellular stimulation of proliferation and inhibition of apoptosis. The more you have of this IGF, we already discussed this in a previous class, the more you have, the more you will have the signaling downstream, and therefore you are blocking the apoptotic pathway and you are inducing the proliferation. So if you have a predisposition to increased glucose levels, in addition to accumulated effects through the fatty acids that are being accumulated, this will lead to increase of the level of insulin for a chronic period of time, and that feeds into the cell directly. The other feeder is that the loss of the continuous induction of the insulin will lead to the reduction of these two insulin growth factor binding proteins, and those will make that guy more available and therefore causes the cell to become more prone to become a tumor. In other words, it's less apoptotic and it's more proliferation. Not necessarily 100% that it goes to a tumor, but it's a good environment that it can become a tumor. Okay. For those who don't remember the IGF-I receptor, we have discussed this in previous slides, in previous classes. I don't recall if it was maybe class 5 or 6. Okay, so this is one of an examples where I'm showing you two factors, which is environmental, which is like diet related, as well as the obesity that could make the environment very well, a good environment for tumor progression and development. Not necessarily it happens, but this is one of the cases that we see. Okay, we'll stop here. We'll take a break for 17 minutes. So we come back at 10. I will be pausing the screen and I will be away for the next 17 minutes. I'll come back at 10, okay? Okay, so we still have lots to go through today. So I will probably go a little bit faster. Now that you know the signaling pathways that we have to discuss, I will go quicker, but I will integrate those in terms of the cancer development. So the next effect we want to discuss is the causes and the promotion of cancer. And here I just give you two other examples, which is the free radical generation and the bystander response. The free radical regeneration, we have discussed that extensively, how it caused DNA damage, genes effect like p53, retinoblastoma, cyclin Ds, and the bystander response is in principle the response that could be circulated from one part of the body to another part. So if, by instance, the head of a person has been under radiation, it will cause the generation of all these signaling molecules. And those signaling molecules will be elevated and therefore could affect multiple tissues. And say, for example, here, spleen is given as an example, that it, oops. That all these generated cytokines or molecules could affect the spleen because it's more prone to cancer development in the body. So in principle, an effect or localized radiation or injury at certain organ that is being responded by another organ, that's because the other organ, which is spleen in this case, as an example, is more prone for tumor development. And the bystander effect is the name of the response we are saying, but it's a signaling molecule, it's interleukins, TNF alpha, it's reactive oxygen species, you name it. I don't know what's happening to my computer today. It's not responding very well. So now that we set the ground for the discussions of cancer development, so how does the cancer develop? So it's not automatic. It's not only one mutation that could cause this cell to directly become cancerous. It's a successive process. And therefore, it might need successive genetic and epigenetic alterations, some of them at the DNA level and some of them are not at the DNA level. And then it has to allow that cell to escape the homeostatic control. And that normally the cell is under control, like P53 and other checkpoints. But now it escapes that control, homeostatic control, and it's able to escape also the suppression of inappropriate proliferation. And that's how it becomes able to survive and proliferate more. So it's a progressive process. The alterations is mostly related to the cell growth, survival, and motility. And this, if we look at the different downstream effects that the cancer cells has to be prepared for, is it has to be generating this tumor environment that we discussed a bit about, and then the angiogenesis, because it needs to get recruited somewhere where it gets energy and gets into the bloodstream where all these nutrients are, and probably later escape and metastasize through the angiogenesis process, as well as the inflammatory response. So it has to be able to kind of escape the inflammatory response, otherwise the immune system is gonna eliminate it. And at certain point, when the tumor is developed, the inflammatory process is not gonna become any important. And I have a couple of examples for you how the immune cells would eventually got tricked that the cancer cells are not anymore considered cells to be eliminated. So there are two sets of genes that you need to know, the proto-oncogenes and the tumor suppressors. The proto-oncogenes, these are normal genes that play a role in the function of the cells. However, when they become mutated or changed or phosphorylated or overly phosphorylated or overly activated, then become oncogenes. So normally when you say the prefix proto, this means these perform regular cellular function, that they become overly hyperactivated, and that's why we call them oncogenes. On the other side, the tumor suppressors, these are genes that would suppress tumors, or in other words, suppress the overgrowth or division and growth of the cancerous cells. So normally the tumor suppressors are the negative regulators, and the oncogenes are the positive regulators of a cancer or a tumor. Between these, there are many, as I said, I don't really have time to cover all the signaling pathways in cancer. So we will focus on the PI3-AKT pathway and the RAS-ERK pathway. Okay. And so if the cells had acquired those characteristics that I just explained, then we have an environment suitable for growing of a tumor. So the 6 major characteristics that the cancer requires are up here. Let's start with this one here, evading apoptosis. So the first thing is that these cells are gonna not respond to apoptotic signaling. So therefore, more survival, more proliferation. They have the self-sufficiency in growth factor. This means those cells are able to produce their own epidermal growth factor and other growth factors, which induces their own growth. They are insensitive to anti-growth signals. So if they receive a signal that blocks cell growth, they don't respond to that. Okay, they have limitless replicative potential. Limitless replicative potential. So this means they can divide for many, many, many times. Okay. Now, the other two is sustained angiogenesis, which means they have the accessibility to the nutrients. So angiogenesis means the generation of these micro endothelial systems, which provides nutrients for the tumor. And at last is the invasion and metastasis. This means they are able to invade other tissues and metastasize from one side to another side of the body. Okay, so these are the six major characteristics, right? But the six major characteristics are caused by what we have discussed in the first 5-6 modules. DNA damage, reactive oxygen species, genetic mutations and aneuploidy, so change in the genetic material, metabolic stress, and hypoxia, loss of oxygen. So all that relates to acquire, so the cancer has to acquire ability to deal with all these conditions. Some of those upper 6 conditions are in fact due to the ability of the cancer cell to escape oxidative stress, to escape DNA damage, to escape, or due to a change in the genetic material, the ability to grow in low hypoxic conditions where no oxygen, or the ability to grow with low ATP. And I will show you one slide that till now we don't understand why cancer uses lactate instead of pyruvate. And you know, lactate is normally generated in low oxygen content, which is a hypoxic condition. Although it doesn't generate too much ATP, but then that's how it uses. And finally, it lives in low pH, and low pH is not a favorable condition for immune responses and immune surveillance. So that's how it tricks the immune system. So for example, one thing we discussed extensively in this class is key genes like the tumor suppressor P53, where mutations alter more than one thing. It alters the angiogenesis and it alters the apoptotic responses. And you can talk about this for like 3 classes on its own. Now, this is not only, I think this slide was from a publication in 2011, and there's more factors that are being added, I think. I will come to this slide next, in a few slides coming down. So we come back to a general description of, well, the cell has the ability to do all these, but how does the cell do all this? And the cell is not like on its own. The cell has contact with other cells and other growth factors and cytokines and milieu and the environment in general. But then we can divide this into 3 microenvironments. Like the first microenvironment here in the circle outside, this is where the cell interacts with other cells. It interacts with the drugs that we probably take. It interacts with a tissue damage that happened in the nearby or inflammatory responses. It interacts with how much metabolites we have and other soluble factors. So this is the first level of microenvironment that the cell interacts with its surrounding. And then all those would feed back into the DNA at the transcriptional level, and this is probably where all our signaling pathways that we discussed play a role, because they hint all the way from the receptor, relate to a second messenger, all the way to the level of the DNA where transcription function is changed, histone methylation is changed, histone deacetylation takes place, or chromatin assembly, and activate or block promoters. And then the third level is going to be how the cell reacts, which is the third environment, which is mostly transcriptional, where the genes has already been changed. Well, you can see here in normal condition, you have this cell where it has lots of gene A, little of gene B, nothing of gene C. But now, after interacting with the environment and relaying those signals to the DNA machineries, then gene A is totally blocked. There's no more generation of the transcript. Gene B, yes, we had it, but now we have tons of it being generated. And gene C, although it was off, now it's turned on. And therefore, the transcriptional profiling of the cell A is totally different from the transcriptional profiling of cell B. And that's why we have now different identities that the cells interact. That's exactly what happens in cancer. It interacts with the environment, integrates new signals, and it changes the identity. So briefly, the two sets of genes that we discussed, proto-oncogenes and the tumor suppressors. I gave you an example here that you really need to remember, the RAS and human epidermal growth factor receptor. These are mostly found to be upregulated or activated in cancers, while on the other side, the tumor suppressors, the p53 and the retinoblastoma, those are the ones that are normally deactivated or inhibited in the cancer progression. I'm sure you remember this scheme because we discussed mutant p53, how it affects the function of the wild type p53. And normally the wild type p53 leads to the activation of the Bax and Puma, which is p53 induced protein that could cause to apoptosis. And on the other side, the p53 blocks the cell cycle P through p21. Now, when you don't have that, or this is mutated, you lose that function. Therefore, you don't have p21 to block the cell arrest, and you don't have the Bax and Puma to induce apoptosis. And therefore, now you have a perfect environment for that cell to grow forever because there's no cell cycle arrest, and never die because there's no apoptosis. On the other hand, you need angiogenesis that is induced by these two proteins, particularly c-MYC is very important here. Okay. More information, but this is a slide you already have seen before, and you have more readings on this, how oncogenes are activated or tumor suppressor become deactivated. Which brings us to that slide that we started with, the question. So particularly, apoptotic signaling has to be turned off. And therefore, in cancer, the levels of the SMAC, which you guys knew it I'm very happy to know that, the level of the SMAC, which is normally released from the mitochondria, are down, as well as the levels of the IAPs are up in cancerous cells. And you can tell that when you have more inhibitors of apoptosis, then you will inhibit caspases, and then you will inhibit apoptosis, right? And therefore, if you have more of those proteins that facilitate the cell death, you could go to apoptotic pathways. So the cancer cells would do all its best to downregulate anything that could cause apoptosis. So it raises this up and brings these down. Okay. Which brings us to the two pathways that we started with. Again, oncogenes are mutated forms of the proto-oncogenes that promote normal cell growth and division. And here, the human epidermal growth factor, the HER2, which is mutated in 30 to 40% of cancers. It leads to the activation of the tyrosine kinases. And now you have the PI3 kinase is active, or the MEK-ERK pathway, which is the MAP kinases activated. Now, this is what we call oncogenic kinases, because once you turn these on, you have the ability to block apoptosis, because PI3 kinase, AKT, blocks the FOXO, as I mentioned. Similarly, ERKs block the FOXO. Now, in addition, the ERKs are more specialized in blocking BEAM, which is a pro-apoptotic protein. And FOXO itself has the ability to induce those two proteins. But now if you block it, either by AKT or by ERK, the signaling from FOXO to PUMA and BEAM is not going to be propagated. So as we have more of these blue or pro-survival proteins, the more blockage on the FOXO will happen, and therefore the more blockage on the generation of the apoptotic proteins, BEAM and PUMA. On the other hand, both ERK and AKT in different ways activate the myeloid cell leukemia 1, which is a BCL2 family that is anti-apoptotic. So you are increasing the level of anti-apoptotic proteins, and you are decreasing the level of the pro-apoptotic proteins in cancer. Now, this is another representation. Probably it's much easier for you to remember. RAS, RAF, ERK, and then downstream signal. And here, PI3 kinase, AKT, and then mTORC. Now, one of the questions that came back before the break is that AKT, it seems to activate many downstream targets. Yes, there's a lot of targets. Maybe the ones that I work in the lab is like 13 or 14 different proteins. So there's tons of these AKT-related proteins. Some of them are direct, some of them are indirect. Now remember, PTEN, it was mentioned in the video, PTEN could block the activation of PI3 kinase. So PTEN is a tumor suppressor, because if we block this guy, we're going to block AKT, and therefore proliferation survival is going to be blocked. Okay. Which brings us to the second molecule that we have to remember, which is the mTOR, or the target of rapamycin, and M is mammalian target of rapamycin. So this is a protein that is downstream AKT. You can see AKT activates mTORC in different ways. What is represented for you here is that AKT block the TSC2, which is the tuberous sclerosis kinase. And normally this guy is a blocker. So you block a blocker. When you block a blocker, this means you cause activation. So again, you get the signal, you activate a PI3 kinase, you activate AKT. AKT, when phosphorylates these two guys, it blocks them. Normally, these two guys are blockers for mTORC. Now, mTORC is active. So the moment AKT becomes active, mTORC is become active because you kill those guys. But how does the activation of mTOR take place? And I named few of those for you. Stress, growth factors, levels of energy, levels of glucose, amino acids, adhesion of cells. And then mTOR is a protein that could bind to different other proteins to make complexes. So there's mTORC1 and mTORC2, which are depending on which proteins bind to mTOR. So if I ask you what's the difference between mTORC1 and mTORC2, it's purely the complex that is being generated. What binds to mTOR defines if it's mTORC1 or mTORC2, which is target of rapamycin complex 1, target of rapamycin complex 2. But no matter what was that signal, once mTOR is activated, so AKT probably is sitting here because growth factors induce AKT and then it's here. Or it could be sitting here. It's a stress that comes to a tyrosine receptor that activates AKT and then mTOR. Once mTOR is active, then it induces messenger RNA translation. And now, remember what you were discussing? You're discussing a cancer cell. Cancer cell, it needs to grow, it needs to divide, it needs all those components. It has to make membranes, it has to make proteins, and it has to make those organelles. Therefore, it needs to translate a lot of those proteins. And it needs the energy for that. Okay, so you increase the level of the translation, and therefore that's how you achieve the growth, the survival, the division, and then the motility. Because remember, one thing that mTOR affects is the cell motility. Okay, so let's look upstream, downstream of mTOR a little bit more. So mTOR balances between the levels of energy, how much we are consuming in the anabolism and how much we are producing in the catabolism. Now, we need high energy, right? So high energy levels induces mTOR and low energy levels inhibit it. So it's very logical because you need energy in order to grow, survive, divide, and all that. If you don't have energy, this guy is going to be locked, is going to be turned off. If you have high level of energy, then you are going to proceed. Okay, so there are intermediates in the cells which look at the levels of the energy. And those could activate or not the tuberous sclerosis complex 1 and 2, TSC1 and TSC2 here. Okay. So if you get growth factors, or whether you get lots of energy or nutrients, all these will block the blocker, and therefore you activate mTOR. Because when you are speaking about levels of growth factors high or nutrients or energy, this means this cell is ready to divide, grow, and survive. Therefore, there's no reason to keep it under blockage. And no matter what is the route, the final component is that you block the blocker and then activate mTOR. Some of these molecules are very well known. This has like probably 13,000 papers about AMPK and metabolism, because it's the sensor of the energy in the cell. So mTOR is getting activated. So what happens next? It has two downstream signals. One that is an inducer, that it phosphorylates the S6 ribosomal protein, and the S6 ribosomal protein is important for generating more ribosomes. The more ribosomes you have, the more proteins you are generating, the more building blocks for your growing cell. The other is an inhibitory effect, and that is over the 4EB binding protein. I have the text for the 4EB and E1F, which is the initiation factor of translation, and how it works. So normally, when mTOR phosphorylates 4EB, then it will block it. Okay, and then that will induce the phosphorylative event of downstream signals, like looking where is the stop codon and initiation of the translation complex. And therefore, if you look, you are generating more ribosomes here, and on the other side, you are generating more messenger RNA being translated to proteins. And both are very important for growth, survival, division, and motility. So the signal that we looked at here that came as a growth factor to the receptor and then induced PI3 kinase and AKT and then mTOR is now translated to all these functions through mTOR. Okay. So the inactive form again of 4EB binding protein is unphosphorylated. The active form is phosphorylated. And then now you can release the cap and proceed with the translation. You can read through this and let me know if you have any questions. Okay. So now we have a cell that get prepared. It got either mutated, levels of oncogenes are high, levels of tumor suppressor is low, growth factors, signaling pathways like PI3 and RAS are active. They start to grow higher and they don't die, so they have less levels of apoptosis. So they got into the level where they have increased growth and decreased apoptosis. Some of these causes could be purely genetic, like it's a mutation that accumulates with time, and it could be at different levels. It could be 1-point mutation, it could be a subtle alteration where you have a piece of the chromosome, like insertions or deletions. You could have a multiple level of chromosomal aneuploidy or heterozygosity that is being lost. You could have an amplification of a certain gene itself, just that this gene caused it was inactive or active to a level 1, as I saw in the identity, and then you boost that activation like 15-fold, 100-folds. You could silence a silencer, and then you can activate those genes. And finally, this is one of the examples you need to remember, at least from this slide, is the exogenous sequences that come from the tumors, from the viruses to tumors, where infectious agent can trigger the tumor itself, like in the condition of the human papillomavirus, because it could promote oncogene activation and would inactivate tumor suppressors. So there are two examples that are very well known from the human papillomaviruses because it generates two different proteins. One of them is called E6. The E6 protein could promote degradation of P53. And the other one is E7, which inactivates retinoblastoma protein and cyclic kinase inhibitors. And if we go back a few slides to the P53, so the E6, it inhibits this guy. So you could tell the effect of the human papillomavirus. Okay. Now, is cancer hereditary or not? It is partial, because itself is not like you are not prone to become like having a cancer right away, but the chances of having a copy of that gene being mutated. So if someone has a mutated gene of breast cancer 1, he has 60%, up to 60% chances that get breast cancer. The chances of acquiring a mutated form of the breast cancer 2 is that 45% that develop breast cancer, compared to 12% in normal people. Now, what makes this up to the 100% is the other factors we mentioned: the diet, the environmental factor, the other genetic dispositions, the reactive oxygen species, the all the components that we discussed before. Okay, which brings me to this gene here. So I will stop a minute and refresh with you and open a stage for a couple of questions before I proceed. And then I will go to describe the KRAS protein and the signaling through KRAS. Because P53 we already discussed a lot. MYC and Notch, I'm not going to cover that. And beta-catenin, although these are very important, but we don't have the time for that. But this already sets the ground for you for signaling pathways that really affects cancer. And I think it will be very easy for you if you read about it in the future, that you can easily integrate how MYC and Notch and beta-catenin affect cancer progression. So you have a question that you didn't get. You have a slide that you want me to go through before I move forward to discuss KRAS and mutations in KRAS. Questions, speculations, anything not clear so far? Am I going so fast? Am I going so slow? Am I going in between? Sounds good. Okay. So the 6 lesions in terms of the genetic, wait, maybe somebody asks something. Can you go over the slide of apoptosis signaling pathway? Margaret, are in terms of cancer, you mean? Or in terms of apoptosis signaling pathway? Because this we are extensively discussed during a previous class. No, I intentionally went fast because we already discussed that, but I will come back if I have time at the end for you, okay? I will come back at the end of the class to discuss that for you. So these 6 different mutations, I gave you an example on the viruses. I'm not going to extend on that more. And gene silencing, methylation, histone modification, and microRNAs, we discussed that for the DNA methylation and histone modifications. We already passed over that somewhere in lecture 4, 5, 6. So I'll give you a couple of examples on point mutations and chromosome changes. So missense mutations. Yes, missense mutations of change of only one amino acid has been reported to cause cancer. And there is a couple of examples for you here. Where oh, I think there's a slide missing. I think there's a slide missing here. Okay. The second one is the chromosomal arrangement. The chromosomal arrangement, it can cause an effect on one gene or multiple genes. So here is a normal situation where you see cells are being grown and gene A and gene B are being transcribed and being translated into their respective proteins. And therefore, the cell is grown normally. When whatever the cause is, if gene rearrangement chromosomal arrangement take place, then if it happens to affect the gene A and B, there could be multiple outcomes. One of these outcomes is represented here for you, where as if a cutting through these genes happened, and then a part of the pink chromosome now is on the blue one, and the other one other way around. And now you have the chromosome rearranged, but unfortunately, both gene A and gene B are kind of disrupted. Or now gene A and gene B are fused into one novel gene, which is gene fusion. And therefore, when you turn on the transcription of that gene, you are going to generate a protein that is fused, because you start that protein being translated from the messenger RNA as one protein only. Now, this fusion protein is not going to perform the function the same way as the gene A, gene B separate. And that's how it could be a reason of cancer progression. And the most known out of the cancers is the Ewing sarcoma. There's another form where alveolar rhabdomyosarcoma, which is normally affecting stem cell factors PAX7. This is a stem cell transcription factor, and FOXO. Again, FOXO is overly activated in this condition. So that's an example of chromosomal rearrangement and how it causes cancer. An example of the exam, yes, I can present this for you, and I can ask you give me an example. Alveolar rhabdomyosarcoma or synovial sarcoma or Ewing sarcoma. Okay. Oncogene activation mechanisms. So this would be the fourth one on the list. Amplification of gene expression. Okay, amplification of gene expression. So we have a gene under a control of that promoter, and this is now, it's called a proto-oncogene. Okay, so it's performing regular function. You got a point mutation. Okay, so this is the slide I was mentioning. It was lost, but apparently I left it here because it summarizes two things at the same time. The point mutation itself, it causes a generation of proto-oncogene to become oncogene, or gene amplification, meaning now you can amplify that gene more than usual. And the third situation would be when you have a chromosomal translocation where it is in fact translocated, instead of being under a well-tied and low-level expressing promoter, becomes under a control of a promoter that is producing tons of this, so it's overexpressing it. The situation become even more dramatic if we got a fusion of that promoter, similar to what I just described to you here. Okay, so those 3 or 4 scenarios could happen just because of instability of the DNA. So you could have a point mutation, and that's happened in a lot of the RAS proteins, and I will give the example of RAS in the next slides. It could be amplification of the cellular gene, where you have higher level of this protein, and not surprised that you have MYC as a protein to be upregulated in neuroblastomas. And the third could be a chromosomal translocation. The easiest form is that you generate a fusion protein, and the more complex is that you have generated a chimeric protein under the control of the different promoter. And there are different examples here. So when you have the chromosomal translocation, the Burkitt lymphoma is the best example of it. because now you have a much stronger promoter. You are generating tons of that gene. Or when you produce novel fusion protein, that's another example for you here, okay? The bottom line is, in such schemes where it's not really very crowded, those probably could be some of the exam questions. So I could ask you, what are the routes that could lead to oncogene activation from a regular proto-oncogene to an oncogene? And be careful when I ask about the genetic basis, this means I'm asking about the point mutations, about the gene amplification, about the chromosomal translocation. I'm not asking about the growth factor related. So you have to be careful how to read the question. And again, next week, I will give you some exam questions, which brings us to the next target we have to read at is the RAS protein. So why RAS protein is very important to know, because these small GTPases, this is a family that comprises many proteins, and they are very important regulators of cell growth and differentiation survival. So when a RAS protein is bound to a GDP, it's inactive. So you can see it's recruited here into a membranous-like, it's a pharyngeal membrane anchor. When it becomes active, it depends on a growth factor receptor that bridges the signal directly to the inactive RAS. So the growth factor came in, growth factor receptor gets activated, could be tyrosine receptor. Then that bridging protein now relays the signal, and the RAS becomes active, because now we get this GDP, which is diphosphate, guanosine diphosphate, into triphosphate. And again, whenever we have the triphosphate, we have one more phosphate, we have more and more energy. And now the RAS protein is active. Okay. Now, activated RAS sends growth signal to the nucleus through cytoplasmic kinases, starting with the activation of RAF. So we started with RAF and MAP kinases, could be PI3 kinases. It might be other kinases. Again, we are focusing on this arm here. Okay, we are focusing on this arm here. So let's imagine a scenario where RAS is mutant, where if you give RAS, the regular normal RAS, if you give it GDP, you take the GDP out and replace it with a GTP, then there is a cycle that could cause the deactivation, and that's what I said. It's mainly depending on proteins called gap. Okay, the proteins gaps. But imagine that this active RAS is mutated, so now it's not responsive to the gap, or in other words, it would not get rid of this GTP. So the mutant RAS protein is permanently activated because it cannot hydrolyze GTP. And therefore, you can lead to a situation where you have a signal that is continuous, but now you are not depending anymore on the growth factor. And that's exactly what happens in many cancers. There's a video here that I really want you to watch about how RAS become active. Let me see how much time we still have. We still have time. I think we could go for that because it's a good my computer today is not fun.
Give yourself 28 days and become the most dangerous person.
Do you guys see the screen? Please confirm.
Yes.
Thank you. Okay, so let's see how KRAS is activated.
Tumours contain lots of different cells. Apart from the cancer cells, a tumour can also include immune cells and stromal fibroblasts. In pancreatic cancer, a protein called KRAS is often mutated. Healthy KRAS occasionally sends a message that tells a cell to divide. When KRAS is mutated, it becomes hyperactive and can't switch off anymore. This causes KRAS to transmit too many growth signals within the cancer cell. This is called cell autonomous signaling. In addition to cell autonomous signaling, mutant KRAS can signal to nearby fibroblasts. Because fibroblasts use different signaling pathways to cancer cells, they interpret and process signals in ways that cancer cells cannot. This allows mutated KRAS in cancer cells to change the behavior of neighboring fibroblast cells. This is called non-cell autonomous signaling. Non-cell autonomous signaling can cause fibroblasts to produce unique signals of their own. These unique signals can then return to the cancer cells. Back in the cancer cells, they activate new signaling pathways that the cancer cells do not activate on their own. Because these signals start with mutant KRAS in the cancer cells, travel through the fibroblasts, and then back to the cancer cells, we call this reciprocal signaling. So mutated KRAS can drive cell autonomous signaling in cancer cells. KRAS also drives non-cell autonomous signaling in the nearby fibroblasts. We now show that mutant KRAS can hijack nearby fibroblasts to drive reciprocal signaling back in the cancer cells. Finding this important new way in which mutated KRAS can promote cancer growth may provide new possibilities for therapeutic intervention.
Main pathway.
Okay. So we got this KRAS, which is formally active, and we got this signal that is continuous to induce the cell growth. So that's exactly how KRAS do that. So this is the sequence of the KRAS in the wild type genes. And that is the only one missense mutation. So it's the 1-point mutation that takes place where G at the location 35 becomes a T or becomes an A. So the KRAS is now called KRAS G12V, glycine 12 into valine. So whenever you read this in a genetic paper, this means the KRAS gene has been mutated at glycine number 12 to become a valine. And the change, if you read it in the text, it's the location of the sequence 35 G into T. Or again, at the DNA sequence 35 G into A. This caused the KRAS mutation into glycine into aspartic acid. Okay, so this is one of the most known. I think this is like probably 51% up to 45%. In that video, they mentioned pancreatic cancer, but in almost any cancer type, there are some sort of mutation that accumulate at certain point with the KRAS. Okay, now recall from the first few lectures, what is the difference between glycine, aspartic acid, and valine? So remember, amino acids, they have the carboxyl group, the amine group. So glycine is the simplest amino acid and the smallest one, in fact, because it only has the hydrogens as the groups around that central carbon. Now, if you look at the aspartic acid and the valine, now you could tell that this is a much more activated form of an amino acid, if I can call it, because you have more oxygen and hydroxyl groups that are prone to perform function. And remember, when we discussed that aspartic acid is probably one of the most common amino acids that is used in the pockets of the enzymes because its ability to perform these electrophile addition and attack. That's probe because it has OHCO on both sides of the amino acid. Okay, so the more you have the ability to perform functions, so here you can tell that this is probably now it's attached by another amino acid and therefore is not able to generate any type of reaction. But if you look at this, one of the hydroxyl group is needed to generate the bond with the next amino acid, but the other one is free as an R group where it can generate the reaction. So that's why when you change glycine into valine or aspartic acid, which valine has the similar effect as well, you might have much more reactivity. And when we perform experiments in the lab where we have an enzyme that is very active, like well, very active. When we have an enzyme that is active and we want to deactivate it, we normally go and mutate aspartic acid in the enzymatic pocket into glycines or alanines, one of the two. So here the cancer cells find a way to get an inactive gene into an active gene. So or inactive form of a protein into an active form of a protein. But this slide is meant to tell you something else, which is and nowadays we have a new system that's called CRISPR-Cas9. And there's lots of efforts that are being made in order to use the CRISPR-Cas9 to correct the mutant RAS in the genes to bring it back to the normal. The way this works, it needs a guide RNA, a guide DNA that normally is used by the CRISPR-Cas complex, CRISPR-Cas complex. Then that guide will cause the change depending on the signal that you introduce. So you would tell the CRISPR-Cas9 where to bind and what nucleotide to change, and it will perform that. And this has been done in dogs already, as I mentioned before. So this is a very good approach that probably in conditions where we have cancer predisposition based on mutations that are on G12 RAS being mutated into valine or aspartic acid, we could correct this back to wild type, and therefore it could be a therapeutic intervention to inhibit cancer progression. Okay, cancer microenvironment, we discussed that, and that video touched already on it, where these cancer cells start interacting and generating signals, and those signals will come back and affect the cancer cells themselves by inducing more growth-related and anti-apoptotic signals that feeds in. Now, at this point, the cancer microenvironment is already being made, and the tumor microenvironment is being prepared to propagate more and more. So probably the cellular aspects, the molecular aspects at the level of the genes that led to the growth, now you have already passed that point, and then you will start looking into a change at the cellular level and the tissue level, where the cells probably get prepared for metastasis. Now, before discussing the metastasis, you have to remember that metastasis is a very, very complicated process, and it's also a risky process for the cancer. So the cancer wouldn't start metastasize unless it has been very well established. So first, it has to transform from the regular cell type to the overly growing cells. Then it has to find the microenvironment with the angiogenesis, as we described, and get the nutrients and the factors that it needs from the neighboring cells and from the blood flow. And after that, it has to invade the capillaries. But now the process becomes really complicated because it has to survive a new environment. Maybe it has created the right environment here, but now as it goes into the bloodstream, it has to survive that transport, and it has to adhere somewhere. So that's a lot of risk that the cancer cells would die. This is a very good risk for our body, in fact, that it could be eliminated. But unfortunately, in many cases, it metastasizes from one side to another side of the tissue. And that's what we call here when it extravasion. So here's you have the invasion where it invades the blood vessels and circulates, and the site where it exits the bloodstream and goes to the next side is called extravasion, where it finally generates new colonies. More videos for you to watch. But at the cellular level, what happens? At the cellular level, metaplasia is the condition we are talking about. Remember, we spoke about smaller size in atrophy, or hypertrophy, increase in size, or hyperplasia, which is an increase in the cell number, meaning it's growing. But now in cancer, there's one more level where it's a replacement of the original cells. Now, these cells, these are normal cells. If they start to grow, this is hyperplasia. But now if they grow and they are replaced by something else, that's called dysplasia. Okay, so this is just different terminology, just to add one more change that happened in that tissue. And finally, the cancer development, the tumor development would reach the level that you have a loss of differentiation or specialization. So if you look at these cells, these are specialized epithelial cells that could perform certain function. but when they become this plastic, most of the time they lose the differentiation, and therefore they don't perform any function. And most of the time, that's when the cancer is ready to get invasive. Okay. A very rough comparison for you between malignant and benign tumor. If you read through it, you will see everything in the malignant tumor is opposite to the benign. Particularly speaking, the mitotic index and the differentiation. So the benign tumors are differentiated, and that's where we lose the differentiation or the function with the malignant tumors. The mitotic index in those guys is much lower compared to the malignant ones. Now, there's nothing complicated. We went through all that. If you read through it and you have any questions, let me know. But one thing I want to add is that in malignant tumor, the cells are not only growing, they grow without growth factors. It's very important to retain this information. They grow without growth factors. And how do we relate this to molecular pathways? We relate it to this guy here, because we don't need a growth factor because we can mutate RAS. Because normally we need a growth factor to activate RAS. Now we don't. If we get this form of RAS, we don't need to activate it. It's already active. And that explains why the benign tumor, the malignant tumors would not need growth factors, and they could evade apoptosis. Okay. Now, if we look on the histology, so these are sections that were taken from the colon, intestine. This is explaining to you what it means in normal, benign, and malignant tumors. So you can see the structure is very well designed, aligned, the acinar cells. And now the structure is still there. You have little outgrowth, but the structure is still there in the benign tumor. But in well-differentiated malignant tumor, then you clearly see that this tissue is totally different from that tissue. Now, malignancy leads to the loss of differentiation. So that means the cells will lose their identity, and they will look totally different. And that's what happened in poorly differentiated malignant tumors. So if you compare a start of a malignant tumor to a stage where it's already prevailing, so it's very well propagated, then you will have very little differentiated cells, and then that affects the structure. So you would not know that this is a colon cell, like section of a colon compared this guy to this guy. And more importantly, if you look at this section E here, where anaplastic malignancy, this means also lost the function and lost the ability to perform any related function to the intestine. Okay, so that's how we look under the microscope when we want to check the cancer cells, because first indication is that there's more cells here than there. Second indication that the cells look smaller, so there is much more, so probably it's in a stage where the growth is uncontrolled. And then the third stage is that when the tumor is undifferentiated, where you can see some sides of the colon looks like normal and the other sides absolutely abnormal. And the fourth stage is that total loss of the function in the tissue. And if we go back, we insist on these couple of changes in terms of the cellular function. So the malignant tumor can divide without growth factors. They can make their own. Oh, that's something else. So they can make their own growth factor. So if they reach to the limit that they need to generate a growth factor, they could generate those growth factors that autonomously affect the cell itself without the need of fibroblasts or other immune cells or whatever. But still, they could feed into the environment the way you saw on the video, that it feeds into the fibroblast and the fibroblast feeds back to the cancer cell. But yes, they have the ability to generate their own growth factors. They could generate their own receptors. And remember from the previous class, if you want to activate a pathway through a hormone, then you generate more receptors for that hormone. Probably I have the slides, yes. So you can upregulate the number of the receptors, and that's how you activate more. So the cancer cells, they have the ability to produce more receptors. So the human epidermal growth factor that is being mutated, you could generate more of those receptors. So you could have more activation through that pathway. Another thing is that more, like, if we want to think about a therapeutic target, is how to generate drugs that could affect those receptors. So we could think about generating antibodies that block those receptors. So in principle, you generate an antibody that blocks the hormone to bind to that receptor. So no matter how many receptors you will have them blocked by antibodies. And then this hormone is not going to be able to signal to the cell. So we come back to these two very important notions as well. Normally, if you grow cells and they become dense to a level in a normal situation, they die. If you they like in the lab, if you put cells in a culture dish and they become 100% confluent, the next day they die, or maybe the day after. The cancer cells, they don't have this problem. So they don't have density dependent and contact inhibition, because they squeeze to each other, it doesn't matter, they still survive it. Normal cells don't. So this is called cell density dependent and contact inhibition. The second thing is that anchorage dependence. The anchorage dependence means the cell has to be in contact with the basement membrane, similar to what I've shown you in the previous slide when we just start discussing in the class, where those cells were relying on those basal membranes. Without those basal membranes, probably the cells will die. And that's because the cells have anchorage dependence. Now, cancer cells, they don't have that. And that gives them the ability to be able to metastasize later on, because they can simply dislodge from the site of the tumor and grow and then go into the bloodstream and invade other tissues. Okay, which brings us to one of the diagnostic tools that we use for cervical cancer. Like this is what's called the pap smear. Normally, this happens when women are pregnant. They take like some pap, and they will take some of the cells, and they will look under the microscope how these look like. This will dictate if the tissue is normal or abnormal. And the point we see it is that if the cells are larger in size, they have darker or irregular nuclei compared to normal cells that they have regular size and they have normal size nuclei. So you could easily pick that these cells are overgrown, they are smaller, they are denser, sorry. They are denser, they have much bigger nuclei. And that's an indication that probable dysplasia in the cervix. So I put for further reading these two slides for inflammation. The bottom line is that although cancer cells are being eliminated every day from our blood and from our body, from our tissues, through our immune system, the lack of the ability of the immune system to get rid of those cancer cells is when the tumor progression takes place. And sometimes the immune system even makes it worse. In another sense, in other words, that the cancer finds way to evade the immune response because it's producing a lot of lactate and acidic conditions around. So that's why immune cells could not really kill the cancer itself, one thing. The other thing is that at certain point, and that's what we see here, recruitment of cells into the tumor could lead, in fact, to deactivation of the tumor response. Because if there is an immune response, it has to stop at certain point. If it has been propagated for a long, long period of time, then the Treg cells will come to the show, the CD4 positive, and they would block the immune response. And that is where the cancer progression become much uncontrolled. So read through these, is for your own knowledge. And I think I will stop here. Probably there's a few slides about the cancer drugs. So I will stop with this slide here. This is the last one. So I listed for you 9 different ways how the cancer cells become resistant to drugs. And based on the discussion that we have today in terms of signaling pathway, in terms of the DNA damage, in terms of the growth responses, you can tell that the cancer cells first decrease the uptake of a drug, or it just shuffles this and pushes it outside of the cell if you give it a drug. It could compartmentalize the drug, so it can sequester it or change it. And changing it means you can metabolize the drug in a different way. Also, it could change the target. You design a drug toward one target, and then it can generate another one that is totally different. And of course, it can increase the DNA repair damage and resistance to apoptosis. So I hope this gave you an overview. I know it's a dense, but unfortunately, I can't make it. We don't have too much time left. If you have questions, please go through these slides and let me know. So probably this is one of the things I can come back next week. I will speak about it in few words here, where CAR T cells, which is chimeric antigen receptors, could be used as a therapy. Because if our immune cells are not able to recognize the cancer cells, what we can do is to take these immune cells from the blood. So we withdraw the blood and we take these T cells. And then we take them to the lab and then we engineer them. We add the DNA response, the DNA gene, like genes that could make the cells recognize the cancerous cells. So therefore, we need to have information about what's the type of the cancer and what's the best antigen to express in those cells. Now, when we bring this back in, this cell is going to be able to recognize the antigen that we have expressed. And now, if we introduce this back to the body, then our immune cells will find a way to recognize the antigen that we are expressing, and therefore it will be recognizing the cancer, because now this is the cancer immunogen. And after that, this cell is going to be able to lead to the killing of the cancer cells. So probably this is the slide I will come back next week. There's a video for you here that explains. It will be my first slide. I will speak like a few minutes about it. Other than that, so I will stop sharing. I will stop recording. Okay, Margaret, could you please stay? I will explain to you the questions for the quiz. Mostly will be multiple choices, but they will be a different level of complexity. Yeah. I will announce, so just I'll keep the recording running. So I will announce more information about the quiz, how long it's gonna be, how many questions, and how many points. Please note that it will be on Brightspace with the respondents lockdown at the end of the next class. Okay. Okay, so I will stop recording right now.